before we get started, somebody apparently purchased 4 tickets and they don't have them in their pocket. They might have left them right up on the...
Yeah, a lot of people would come in.
Yeah, Dean said it was one of the last people in. They were on the table, on the sign-in table.
Probably mine.
I got 4 tickets.
Do you have them in your pocket? Well, here they are. All right, we got that settled. We got a good lineup today. Guest speakers. Bill Ardren's going to lead it off. He's been here before talking about the salmon genetics program that he's working on. And Bill is going to be followed by Tori Pinero from UVM, grad student who's working on a lake trout telemetry project with Ellen. And we're going to take a break after that, at which point we're going to do the giveaways, a little twist this time. And it'll be followed up by Matt Tremblay, who's going to be talking about the Lake Champlain salmon fishing techniques. So anyway, let's get this rolling, and here's Bill Ardren.
Thank you very much, Bill. It's nice to be back here because what I'm talking about today is our results of scale samples that you sent in. So we came here 2 years ago, Andrew Harbeck and I, and we passed around a cardboard cutout of a salmon showing what an LV clip looked like, and we said we'd love it if you could take some of these scale envelopes and send them back in if you catch one of those LV clip fish. And so today what I'm going to do is tell you a little bit about what we found so far with those samples that you sent in to us. So thanks again for helping us out with this. Andrew wished that he could be here. He's trying to finish up his PhD right now and just couldn't get away, so I'm going to try to fill in the best I can for him. So when we talked about that angler sampler program, this is kind of the citizen science idea, right? That we've got folks out there handling fish. Is there a way that we can collect information on those fish to maybe make the fishery even better out in the lake? So we went through and talked about what LV clipped fish were, how to collect the scale samples, and what they're going to tell us, and then I'm going to give you some of those preliminary results. So we all know, and a big reason that this room fills up a couple times each spring is that Lake Champlain does have a really good Atlantic salmon fishery right now. And despite over about 70,000 smolt stocked into these key tributaries each year, the return rates could be, increased back to the rivers. This is one that Andrew wrote, so it's actually not quite accurate because I know, for example, and I know Brian Chipman back there is thinking to himself too, that it's more like about 30,000 or 300,000 smolts that go into the lake each year. But I think he was trying to emphasize those that go into a couple key tributaries. And what we see is, coming back to the rivers 2013, 2014, around 150 to the Winooski, somewhere around 70 to 100 back to the Boquete River. But we know there's a lot more salmon out there in the lake. So how do we improve the river fishery while maintaining the lake fishery? Yeah. Sure, thank you. So while he's doing that real quick, the idea behind this study is kind of twofold. One is, how do we get more fish back to the rivers to enhance the river fishery? And then if we do that, how do we also take a look at what's going on with the lake fishery at the same time? Thank you. So the idea behind trying to improve the in-river fishery is to improve imprinting to the rivers. And we know that imprinting tends to occur from the parr to smolt transformation. So that parr form that you see over there with the parr marks on it before it turns all silvery to head out to the lake is the form that is in the river. And we're stocking them in essentially right before they go through that parr to smolt transformation. And we're hoping that they're going to remember the river and come back as adults. In the Winooski River, the hatcheries were rearing them a couple different ways, and we were interested to know if the fish that were reared on well water their whole life return back better than those fish that were reared on natural temperature water and natural scents in the water, meaning the brook water, Furnace Brook, down at Eisenhower National Fish Hatchery. So that was the first experiment. We put in half the smolt stocked into the Winooski were reared on well water. The well water has this advantage. It has two advantages. One advantage is it's disease-free because there's no other fish in it, so you don't have to worry about the fish getting any diseases. And the other advantage on the well water is it's nice and warm through the winter, although this winter it didn't help out as much. But a lot of winters, those fish that were on the brook water, you couldn't even feed them. The raceways had a layer of ice, right over the top of them. So it was a more natural setup for those fish, but they were a little bit smaller, and they had a little bit higher chance for maybe some disease issues. And that's why folks in the past had used, the well water more often than the brook water. But we were interested in knowing if you could get this brook water to increase the numbers that would come back to the rivers. And then in the Boquet River, we used the well water reared fish, but we just asked the question, hey, maybe if we put them out in the river a little earlier in March, and by the way, we struggled to put them out in March for a couple of years because there was all kinds of ice on the river, and as I was driving over today, I was thinking to myself, boy, we wish we would have had it going this year as well. But we'd get them out as soon as we could in March, and then we'd stock the other half of them out in May. And that's when New York stocks most of their smolts into the river. The idea there was if we got them out a little earlier, we'd make sure they went through that part of smolt transformation in the river. If you get them out in May, maybe they're already smolts and maybe they won't remember so well. So in order to do this study, we need to be able to identify the fish that are part of it. And what we did is we clipped the LV fin, the left ventral fin, on all the fish that were part of it. And we stocked them into the river system. It was about a third of the fish that went into Lake Champlain for those 3 years. And then the step 2 was to ID the experimental group, when and where it was stocked. And for that we used genetic tags. So for the study we had a 2010, 2011, and 2012 year class. We then outplanted them as smolts, that part of smolt stage in 2012, 13, and 14. So total we had approximately 90,000 smolts that went out into the Winooski, and we had about 135,000 smolts over those 3 years that went out into the Boquet River. Just as a quick reminder, that left ventral fin, and it's the fish's left, as Andrew would always say, is located right down there on the bottom of the fish. And this was just a little bit of a reminder of what folks did. When they caught a fish with that LV clip, they were taking a scale sample from it, sending it in to us, so that we could use the genetics to tell them where it came from. So in the rivers, the Boquet and the Winooski, it isn't such a big deal for us to catch the fish because there are fish traps that are there. Now that isn't true anymore in the Boquet because they actually removed the Willsboro Dam in the Boquet River, but we found other ways to catch those with trap nets and with what we're calling kind of a tooth net or real quick set gill net that we can catch them and release them alive. And so catching them in the river hasn't been a big issue for us, but getting them in the lake, that's tough. But all the boats in here, all the people who fish in here, has been a huge help in helping us get a hold of those fish out in the lake. So this is what anglers use. This is what folks used out there in the lake. We handed out those envelopes, and we asked folks to take a few scales off the top of the fish right behind the adipose fin there, and make sure that left ventral fin was missing and that the adipose fin was also present. That adipose fin's that kind of lumpy fin, on the back of the fish right behind the dorsal. And that was important because the state of Vermont had another study going that they clipped the LV fin and the adipose. So you had to make sure that adipose was on there. And I was very I guess I wasn't surprised, but I was, it made me pretty happy to see that, you know, folks in this room really were able to take that to heart and did a good job identifying. You'll see that later. Then the scale samples would come in with all the information written on them. All the scale samples that came in were able to be analyzed with genetics. The important part, if we ever do this again, and this only happened on a couple scale samples, and you know, I know it would be an easy thing to make a mistake on, is we gave out all the scale samples in Ziploc bags, and every once in a while somebody would catch, you know, a fish, take the scale samples off, and you know, the envelope would get wet, they'd zip it right back up, and then it would get a little bit moldy, you know, in that bag before they would send it back to us. So it is important to try to, you know, leave them out, to let them air dry first, and then you can throw them back into the Ziploc bag. But even this one that had a little bit of mold on it, you know, still was able to give us good usable DNA, which was great. On the back of the directions, we broke up the lake into different sections. We had one section which was the inland sea, another one Mallett's Bay, another one Main Lake, another one Converse Bay, Arnold's Bay, and South Lake. And so we asked folks to mark down on the front of that envelope what area they caught the fish from. And that gave us an idea of, you know, the distribution of where these samples came from. So, the genetic tags, how did we do that? Well, we went to the hatchery at the time all the fish were produced for the smolts that were stocked, and we took a genetic fin clip from each of the parents that were crossed, the male and the female. And then we just used that information to be able to identify. So you can see there, female 1 was crossed with male 1 and female 2 with male 2, producing the progeny. You LV clip each one of them, you can assign it back to its parents based on its genetics, just a pedigree reassignment. It's the exact same stuff if you've seen CSI or all those kinds of things where they go out and they take the blood sample and they take it to the lab and they run the genetic analysis with their peaks. That's the exact same technology they use too to be able to ID fish coming from a certain family, so that they share the same genetic markers with their parents. So why go through all this effort? We're trying to figure out, will changes in the hatchery rearing or time of stocking impact the adults caught in the lake fishery and the number of returning adults to the river? So I'm going to just say this real quick just to make sure that people understand, you know, how this is beneficial to us for trying to enhance the fishery. If we have fish that are better able to imprint on the river, then those fish could come back at higher numbers back to the river and help us create, well, not create, but enhance, you know, a river fishery that is easily accessible to lots of folks, you know, in this room too, and might be, you know, a really good fishery to get a lot of people hooked on salmon at first as well. The other thing about this is that these fish, if you're going to come back to the river better, maybe that means that you hang out in the river longer, which means maybe that not as many make it to the lake because they get eaten by walleye or other things along those lines, or they get eaten by gulls. The other thing that could happen is all the fish could go out to the lake from one group and hardly any of them come back. So they contribute to the lake fishery, which is great, but they don't contribute to the river fishery at all. So that's why we kind of wanted to figure out and go out into the lake, you know, what was out there in the lake. So did everything make it to the lake, or only some of them? Or was there a whole bunch in the lake that never made it back to the river, that kind of stuff. So getting samples from the lake and from the river helps us tease out those things. So we got a total of 150 samples back, with 89 that were submitted in 2014 and 61 in 2015. Thank you very much. That's a huge sample. That would have cost a lot of money if for the federal government or state to go out and try to angle or trawl or gill net or trap to get that many fish. So that, this is really, really nice and really, really helpful and a nice large sample size, so thank you very much. If Andrew was here, he'd be thanking you a lot as well. 103 of those salmon we've been able to assign back with confidence with the genetic tags to those different families or those different groups. So out of 150, some of those fish Andrew's still going through the quality control on, so he needs to run some additional tests to make sure that they are assigning back or they are not. So I anticipate that number will go up slightly. But some of the fish will never be assigned back because somebody just made a mistake in the fin clip ID, or somebody made a mistake when they clipped the fish, too. So all those sorts of things can happen. So I would have been a little suspicious if Andrew would have told me he's assigned all 150 back, because I think there's going to be some error in there, both on the angler part, there's going to be some error on the analysis, and there's going to be some error on the, when the people clip the fish as well. But 103 is great. So of those 103, we were able to assign 64 back from what came back in 2014 and 39 that came back from 2015. So what strategy's working? Well, most of the fish that were caught were stocked into the bouquet in May, that later time period. So half of the fish that were caught out in the lake in terms of those smolts that were outplanted from the two rivers were stocked into the bouquet in May. And about a quarter of them were stocked in the bouquet in March. And the Winooski, you could see a huge difference there, right? Those that were reared on the brookwater tended to, looks like, get out in the lake better than those that were reared on the well water, and be able to be captured in the fishery. So that already tells us a lot right there, right? So in other words, in the Winooski, we've already switched to a more natural system where we're not using a well water reared fish. We're using either a lake water reared fish or a brookwater reared fish, the more natural temperature regime. And then here in the bouquet, we've already switched over to the May stocking component as well. But we mixed it up a little bit here. We not only did the May stocking in the bouquet, but we also used the brookwater. So we tried to even push it up a little higher, right? Because it looked like that brookwater did a lot better, with 22 fish caught out in the lake as opposed to only 5 that were on the well water. Yeah?
Did you track the samplings to see where the fish were caught based on those numbers?
Yep, that's coming up on the next slide, so great, good question. So where did the samples come from? The samples came from throughout the lake, although Converse Bay, it doesn't really match up totally here, so you'll have to bear with me a little bit in terms of the colors. Converse Bay had about 34, well, it had 34 of the fish, had the largest portion of what we had. The main lake here, this is not a surprise probably to most people in the room, right? In terms of the deep part of the lake and the best salmon fishing, as well throughout. So most of the samples came from Converse and the main lake. We had the fewest samples came from Mallett's Bay. We only had 1 fish from there. And the South Lake, we had 6 fish from there and about 18 from Arnold's Bay. And we did not have any. I was thinking about this as I was driving over, as I reviewed what Andrew had sent on. We didn't have any samples that he sent me in the spreadsheet from the inland sea. Is there somebody in the audience here who knows they caught an inland sea fish that they sent it in? No? Okay.
Caught plenty of IS fish, but never caught one with an LV clip.
Yeah. Yeah, it could be that the fish stock that migrate out of the Winooski and the bouquet, it's not very easy for them to get into the inland sea, is it? So it may be that there just aren't that many LV fish even there. So people were fishing for them, but they just weren't catching those LV only fish. So the next step that Andrew's working on right now, and I think this is what you're mostly asking about or interested in, because he showed this last year, but he hasn't had time to break this out yet. Within Converse Bay, right? are they mostly Boquet River or Winooski, right? Is that kind of what you were thinking? You know, and then within the main lake, you know, same thing, is it mostly Boquet or is it mostly Winooski? You know, or were most of those Winooski fish, you know, caught, you know, in the main lake here, and hardly any of them down in this area? That's the next piece that Andrew is gonna work on. And I'm gonna try to talk him into coming next year when the whole thing's put together, you know, both with the river results and with the lake results, you know, to be able to give you the full story here. But he literally pulled these data, he gave them to me yesterday. So he worked hard to make sure he had some preliminary data here, you know, to be able to look at and to be able to thank you. So for closing remarks, these samples that were sent to us to the U.S. Fish and Wildlife Service were a huge help. And they are helping us improve the fishery, you know, with the idea of how we rear the fish and when we stock them. It looks like, you know, by using this kind of adaptive management, and we can, you know, learn from, you know, our results and learn from what you're catching out there to allow us to get more fish hopefully back to the rivers and more fish in, you know, into the boats as well and in the lake. And then finally, Andrew has promised as part of his PhD work, he's got this website which we'll post on the forum. So if you, once it's ready, if you send in any samples, what Andrew's doing is he's making a spreadsheet that you would be able to go down and find your initials, click on it, and then it would pull up for you, you know, on the screen, all the fish you caught, and then how old they were, you know, where they were stocked, when they were stocked, what type they were. All that information would be there, you know, for you to be able to pull down. And if it turns out that you don't have good access to a computer or something too, you could always let Bill or Rob Thorne know about it as well, and I'd be happy just to print it out and give it to those guys to pass on to you as well. So, you know, whichever way will work best for you, but Andrew's done with his PhD in December, so I promise it'll be done by December. You know, hopefully it'll be done even a little bit earlier, you know, than that. He's really sorry he couldn't be here. We do really appreciate all this, you know, work that's been done to send those samples in, and it's really, you know, telling us a lot. So with that, I'd like to end it and see if folks have any questions.
With this type of citizen science thing where you don't have standard sampling, how do you account for uneven fishing effort and how that contributes to your distributions?
So that's a good question. And it sounds like a graduate student asking that question. Right? So what we're trying to do with this type of sampling, it is opportunistic sampling. And we're just treating this sampling by, you know, a snapshot of what's going on in the lake in terms of the abundance of these four kinds of fish in the lake, you know, during those two fishing seasons. Now, if I had to kind of speculate a little bit, is it possible that Winooski fish maybe utilized an area of the lake that the bowcat fish didn't? And maybe people don't fish for them? And I think maybe some savvy people who fish in here were trying to get at that, you know, are there certain, you know, where are those Winooski fish? And, you know, maybe if I fish over here, I'll catch some more of them. It's possible that you know, could be the case, that we could see some of that. So that'll always be a caveat with it. And probably what we should do is some acoustic tagging of some of those fish, which I think we'll hear about next. And so some of that acoustic tagging of those smolts would give us real-time data on where they live, you know, their entire life throughout the lake. And my guess is that they're moving around quite a bit, but apparently not very much up into the inland sea, you know, because we didn't get very many samples there, and it sounds like people fish there, but good question, Tori.
Yeah, that's what I was going to ask about the inland sea, because you don't do any sampling like that, like you did in the main lake in the inland sea. Does that mean you stock bigger salmon in the inland sea so they're more mature?
In the inland sea, and Brian maybe help me out here a little bit, most of those fish would come out of the Lamoille for the most part, you think, or? I'm asking Brian Chipman back here.
Salmon stocked in the inland sea are actually stocked in the lake, because there are no major tributaries in that portion. We do stock them in the Memorial River as well. But again, there weren't any of those specific fin clips stocked up there either.
So does that mean the numbers are down in the inland sea compared to the main lake? or-
No, it just for this study.
I see.
And so we only asked anglers to send us the ones that had the LV clips. Then they were stocked more into areas in the main lake. So to get up over into the inland sea, they'd either have to swim through that small causeway or way up and around. And so it looks like they're not migrating around that much within the system. Oh, I just wanted to say one more thing to Tori, because I thought of one more thing that I think would help with some of the bias that she might be thinking about, is that we do have temporal sampling. So I think it'd be interesting to have Andrew look at data from both of the years too, to see how consistent it is as well. So if we see a consistent pattern across 2 years, that might give us an indication that the sampling is maybe fairly representative or consistent, with some movements. Yeah?
What's the percentage of salmon stocked in the Winooski versus a bouquet?
So over this study, it was about 140,000 smolts went into the bouquet over the 3 years, and about 90,000 or so went into the Winooski.
So you would have expected less out of Winooski.
That's right. So if we would do a statistical test, which Andrew will do as part of his thesis, he could kind of weight that, right? Where he would say, I might expect, 25% less or 10% less for that. But the fact that they were only accounted for 25% of the total adults, it seems to me just intuitively to be less than we would expect.
Wouldn't we also expect water quality in the bouquet to be better than the Winooski River?
I'm not sure
Without
if
the industry around it?
Yeah, so there's industry, there's water treatment plants, although there's a water treatment plant for Willsboro at the mouth of the Bouquet as well. In their, but the upper part of that watershed's up in the Adirondacks in the Bouquet. The fish aren't spending that much time in the Winooski or the Bouquet after they're released. They're usually out of there within a week or 2 weeks. So I wouldn't anticipate that water quality would be a huge component in terms of survival. But I think one thing that could be going on is the Winooski has a big walleye run in the spring, and that'll come up, and we're stocking smolts right on top of that. And they've got to go down through all those walleye that are coming off the spawn too. And so, some of those fish, that we radio tagged a few of those fish, and an angler caught a fish and cleaned it, and it had a smolt in it with a radio tag. And so we do know that that's a source of mortality. To what extent, we're not sure. And I don't think that that's as big a factor, in the Bouquet system, because once they get down into the area where there even could be any kind of walleye component, I don't think there's much of a walleye spawning run there. Are you aware of one, Brian?
Not in the Bouquet.
Yeah, not in the Bouquet. That it isn't even very far for them to get down compared to the Winooski.
So wouldn't you be better off to stock those Winooski fish in Lake Lewis Creek or one of the other creeks?
Well,
that
the
doesn't
nice
have
thing
a
about
walleye run?
the Winooski though is once you get up above the 3 dams in the hydropower, as part of their relicensing to run those dams, they're operating a trap and truck, operation. So they take those 150 fish each year, or hopefully more, coming back soon. They take those fish and they drive them up above Essex 19 there, and they release them, pretty close to the interstate. And then those fish are swimming up, up to the Huntington River and using that upper Winooski as a spot where they can reproduce and spawn. And then that's also a nice access spot for people to fish too.
Like, I don't know if you're familiar with Texas Hill Road.
Yep,
I was fishing in that brook on Texas Hill a couple of years ago. Some of your par were in that brook, way
Right.
up on top of the mountain up there.
yeah. Yeah, that doesn't surprise me. That's a spot that they do quite a bit of stocking because it's also a good nursery habitat. to produce quite a few fish to be able to come down and return as adults. So
I
that,
didn't know if you would stock them there, if they'd come up the brook that far, or.
no, this would probably be one of the first years that we would have natural reproduction in the system, and we are stocking in a lot of those areas in the Huntington below the gorge and above the gorge area there. I keep looking back to Brian Chipman. Feel free, Brian, to jump in and correct me if I'm
Yeah,
Did you say anything else?
The small fry, an inch long or so in the Huntington River, way up into the Hanksville area, and they do come up into those little side tributaries
Yeah.
as well, during the couple of years that they're living in the river before they mature to a smolt and migrate down.
Especially during the summer times too, right, is
Yeah.
kind of a thermal refugio. Yeah, for 'em. Thanks for the question. I thought I saw another one over here, yeah.
Any follow-on studies planned?
Yes, there are some follow-up studies that we're doing right now. We're trying to find right now the return rate back to the dams. So Andrew's working on that aspect of it. So what's coming back to the river with the various types. And now we're trying to study, once those fish are moved over the dams, or in the case of the Boquette, they're starting to be able to make it up over where the dam used to be in Willsboro and move up into about 40 miles of habitat up there. We're trying to document how well they're able to reproduce naturally, and if they're able to produce progeny that can survive, in the system. So we're trying to look at what habitat they use, figure out if we need to improve any of that kind of habitat, and then just document if there's any natural reproduction going on. So that work's ongoing with both states and with, with Concordia University and with U.S. Fish and Wildlife Service right now.
Nice.
Yeah, hey, Bill.
Yeah, Bill, I might have missed it, but are you looking for more samples, you and Andrew, for this season?
Well, Andrew's all done, and he's the guy with the genetics lab, but what we are considering trying to do is there should be a few fish that would be essentially 3 summer fish. So fish that were stocked 2014, they were in the lake that summer. They were probably about 11-inch fish by the end of that summer. Then the following summer, where most of these came in from 2015, right? They would have been 2 summer fish. They probably would have been 16, 18 inches. Is that about right, Brian? And then,
Or a little more.
or maybe a little more. Yeah, and then there are those few that are those 21 plus, right? That people really like to catch. That would be that 3rd summer fish. So I anticipate that there could be a few still out there. And I think it might be a good idea. Maybe I'll get back to you, Bill, and we could post on the forum. It seemed like there was a core group of people. There was about 10 people who submitted most of these samples. And maybe I could just contact those folks, give them the kits, and then if anybody else is interested, you could post on the forum and we could get you some additional scale samples. I don't anticipate it's going to be that many that we're going to get, but I think it would help round out the study.
I have a question on the stocking program for the inland sea. If I understood what you said earlier, there's no natural tributaries that you're stocking. How do you stock that, or do you stock it at all?
So, Brian, did you hear that question? The question was maybe a little more detail on the inland sea, and you mentioned that some fish are stocked into the Lamoille River, and some fish are stocked directly into the inland sea, into open water. Do you happen to know off the top of your head how many?
There's roughly 60,000 salmon smolts that are stocked in the inland sea itself. Another 20,000 in the Missisquoi River, and another 20,000 in the Lamoille River in Mountain Bay.
Did you hear that?
Yep.
Okay.
Thank you.
Yep, you're welcome. Thank you, Brian. I should take Brian everywhere I go whenever I give a talk, I think. Yeah, good.
Did this study tell you why? Because I noticed that the well, the well fish didn't do as well.
Yeah.
And the ones that, were put out in March didn't do as well as the one in May. Are these strictly a numbers thing, or do we have reasons as to why that's going on?
So there are a couple of students that are working on that question right now. And it's a question that people have wondered for a long time. How do salmon find their way back to these rivers? And we're still not really sure how they do that. And we believe that they can use the magnetic field to be able to know where they are. So when a fish is off the coast of Greenland and it needs to, an Atlantic salmon's off the coast of Greenland and it needs to come all the way back to Maine, it uses the magnetic field to be able to sense where Maine is and be able to swim back. And that magnetic field is actually present in these little bulbs that sit right in the front of the head. You know where the nares are, right on the front, and they're paired right there that they have. If you dissect out right in that spot, there's these things that they call rosettes. They look like roses, with all the folds on them, and that's where all the water rushes through, and a fish smells all that water. It has all that surface area in there to be able to smell, and that's how they can smell so well. Well, it turns out that in addition to being able to smell well, if you look at it, it looks like somebody shook a whole bunch of pepper on it. And the reason is that they have special cells in there that sequester the iron. And so there's a whole bunch of iron in those little rosettes that sit on both sides. And that iron that they have allows them to swim through the water and sense that magnetic field and know which way to go. The issue that goes on once they get close is that the magnetic field's not really that sensitive, fine scale enough, that once you get, within a couple kilometers of where you want to be, it can't really tell apart as well. So they think they start using smell at that point. And so we think that landlocked salmon like this are mostly using smell, to be able to smell the different rivers. And what it is they're using for scent to differentiate these rivers is something that's still up in the air a little bit. People think that it is what they call dissolved-free amino acids. So you pick up a rock and it's slimy, right? It's got a biofilm on it. They think that slimy biofilm has unique bacteria and all that kind of stuff that's growing on it, and that when those cells kind of break open and die, and they give off these amino acids, and that every river might have a unique scent, based on the films around the rocks that they have, and that salmon could use those to differentiate the rivers, and so a lot of this stuff has been studied, for, well over 100 years, basically, trying to figure out how salmon home back. Everything I've told you so far is hypothesized, is supported by some experiments, but it hasn't been definitively shown, which ones they use. So it is something that we're trying now. We have a student from Boston University who is trying to understand and do what he's called odor exposures. So he's putting fish in well water, and they think when they hatch out and they kind of come out of the gravel, that they can remember the odor, that's in the river when that happens. So what he's doing is he's putting them on well water where there's no odor, and he's sprinkling in some of that amino acid into half of them, and the other half he's not. And then he's doing these tests to see if they prefer, the ones with the water with the amino acid in it versus not having the amino acid. And so that's one of the things we're testing, is that the well water doesn't have the amino acids. We already know it doesn't have them, but does that mean that, does that make them less able to remember? That's kind of what we're thinking. It's a long answer, but the reason I did that is I think it's important for folks to know that how salmon home back is complicated, and it takes multiple steps. They're using magnetic field, they're using their smell, and those kinds of things. And the mechanism by which well water versus brook water might be that if they don't have that scent when they're growing up, maybe they're not very able to recognize it when we outplant them, into the rivers before they swim out. So if I had to hypothesize, that's what I think would probably be the difference. Yeah?
I know the study's focused on the returns to the rivers, but are the results somewhat indicative of the general survival rate of salmon and how they're stocked in the lake itself?
Well, I think the return rates back to the rivers are probably going to be our best indication, in the end, in terms of survival, from outplanting to coming back. Because the other thing we do is we have those fin clips, Brian and his crew collect fish throughout the lake as well at other locations. And we saw very few of those LV fin clips anywhere else except the Bocat and Winooski when it was time for them to come back and spawn. So we know that they were homing back pretty well, to the rivers, which was really good news. And we'll be able to calculate some survival rates that way. The idea of, what the fishing effort was and those kinds of things, I keep thinking of answers to Tori's question, and the other one that I think we might be able to get at is Brian has done, the state of Vermont is doing an angler creel, which I think many people around this room maybe have participated in on the lake. And that does give us some indications of effort, in the different parts of the lake. So I guess if we assume that the folks that are sending the samples back are representative of that effort that was out there, we might be able to make kind of a back of the envelope, calculation. So are you thinking that it takes 100 smolts to create a fish that somebody could catch in the lake? Is that?
Well, no, I mean, these were tagged fish, so you're looking at that
Yep.
sampling. I'm talking about the general stocking of salmon in general in the greater lake.
Okay,
Does it have a real important information in terms of how you stock them?
yes.
I mean, if stocking, for example, out of the streams versus the well water is much more effective, or stocking them out of
Right.
the Bouquet, maybe that's how the rest of them should be stocked as well.
The one thing I've come to find out as a biologist starting to work with hatcheries in my career, and I've been doing it for about 10 years now, is hatcheries are unique. Each hatchery is fairly unique in terms of its water source and in terms of how it's set up for infrastructure, those kinds of things. So for example, at this Eisenhower hatchery where we conducted this study, they stock about 1/3 of the smolts into the lake. They have both a well water source and a brook water source, that they could use. But the hatchery at Grand Isle, which is another large hatchery that produces at least 1/3 of the fish that go in, all of its water source is from the lake. So they have a deep water and a shallower water, so that they can control some temperature there. So they can only do lake water. They couldn't do well water even if they wanted to, or brook water. And then Adirondack hatchery mostly has some well water and some brook water options. So the ability to be able to translate this to each of our facilities is limited in terms of the idea of using the different water sources in some cases. Adirondack could switch over and start using more brook water, but Edweed couldn't. But the timing of stocking one could be one that we could implement more. And that's one where most of the agencies are stocking in May or April. And I think what we're finding there is if you stock them too early in the system, it's looking like maybe the survival is lower getting out into the lake, as well. It could be a big component of it. So they have to deal with, ice jams, they have to deal with predators longer in the river, those sorts of issues too. So that one might be more transferable, and some of the folks might consider stocking their fish a little bit later because it looks like they can still remember, how to come back even in May. Is that it?
That it?
Well, thanks again, everybody. I really appreciate it.
Thanks, Bill, that was just real good stuff. Yeah, before Tori gets up, I just wanted to mention that Brian Chipman from the State Fish and Game Department, our local biologist on the lake, is here, and he's available for any questions. Right, Brian?
Sure.
Has there been a 14-pound salmon taken out of the Huntington River yet?
Not that I'm aware of.
Okay.
We
And,
didn't send any up that big this last fall. No.
not this year, last year,
2 years ago.
2 years ago. And Mike Wachowski from the state also is here to field any questions, and Mike is the man to talk to about any questions concerning the boat accesses on the lake. So he's here, Tori's going to talk, and we'll have a break, and you can fire away at Mike and Brian. Tori? Sure.
everyone. Really excited to be here. Thank you so much for your time. I've never presented to a group like this, so this is super exciting. And hopefully my PowerPoint works.
Charts. Don't do that.
Actually, I don't. Perfect river right there. But they found one of their own sea lions in November. I think they said it was 14 pounds.